Hello everyone, welcome back to this channel. In the previous video, we have discussed the syllabus of Pharmacognosy and Phytochemistry 1. And in today's class, we'll be seeing Unit 1, Introduction to Pharmacognosy. Contents: definition, history, scope and development of pharmacognosy, sources of drugs such as plants, animals, marine and tissue culture, organized drugs, unorganized drugs, dried latex, dried juice, dried extracts, gums and mucilages, oleoresins and oleogum resins. Definition: pharmacognosy is the study of crude drugs of animal, vegetable, mineral origin and marine sources. History: pharmacognosy historical background is obscure and related to several other disciplines of science, notably botanical, medical and chemical sciences. For better understanding, here I have written in the boxes such as Hippocrates, he is regarded as father of medicine. Aristotle, he is the father of natural chemistry. In competitive exams, many a times they have asked about the scientists and the work they have done. For example, who is the father of medicine? Hippocrates. Theophrastus, he is considered as the father of botanical sciences. Following, there are different scientists and their works done. In India, Charaka Samhita, the oldest classical text in the Ayurveda, dates 200 AD. It is a collection of knowledge of science which describes the chapter on anatomy, physiology, pathology and prescription to cure diseases. Claudius Galen, he was considered as the first pharmacist of the world and also the father of experimental physiology. The pioneering work of Linnaeus, a Swedish botanist, introduced a binomial system of nomenclature. Many times they have asked this question. Pharmacognosy was the first term coined by which scientist? The answer is Sidler and the year is 1850. There are other different scientists who have carried different experiments. For example, Sir Turner, who has carried out the work on mechanic acid present in the opium. And there are different examples as well, such as Albert Neyman, who has worked and carried on experiment on cocaine from coca species. Scope of pharmacognosy. In today's modern world, pharmacognosy has different scope. India also has a diverse cultural heritage due to its biodiversity, though at present Indian healthcare delivery consists of both traditional and modern system of medicines, such as both organized traditional system of medicine like Ayurveda, Siddha, Unani and unorganized system like folk medicines have been flourishing well. Ayurveda and Siddha are of Indian origin and account for about 60% of the healthcare delivery in general, and 85% of Indian population depends on these traditional systems. It is estimated that regularly 1500 to 1600 plant species in Ayurveda and 1200 to 1500 plant species in Siddha have been used for drug preparation. In Indian folk of medicine use, about 7500 to 9000 plant species are recorded as medicinal plants. Here there are different statistics given for the Germany, Italy, France, how the pharmacognosy has its variety application as well and what are the different scope. Now we'll see what is the development of pharmacognosy and its application. First is plant bioactive extraction and isolation. Here, bioactive isolated from leaf, stem, root, bark, flower, fruit, seed or other materials are often identified using a bioassay guided approach. Second is development of plant biomarkers. Here you can see here the DNA fingerprinting technique they have used for the PCR technique, and for example curcumin isolated from Curcuma longa rhizome, which is recently under clinical trial for curing head and neck squamous cell carcinoma, and there are different other two examples as well. Third is development of nanofertilizers and nanomedicine. Nanotechnology in drug delivery is exemplified by nanocrystal, liposomes, nanoparticle protein conjugates, magnetic nanoparticles, nanogel and biodegradable nanoparticles, and also it is also used in the nutraceutical towards biochemical mechanism of healthy aging. Now we come to the second topic, sources of drugs such as plants, animals, marine and tissue culture. Here you can see that plant sources is the odd source of the drugs. Whole parts of the plants are used such as leaves, stem, bark, fruits, roots, etc. Source of leaves. For example, senna is given, and biological source of senna is Cassia angustifolia, and it belongs to the family Leguminaceae. Chemical constituents include sennoside A, B, C, and D. Uses are laxative. Following are the other example of the leaves such as digitalis, eucalyptus, belladonna. So next source is flowers, clove, vinca, hibiscus, and respective biological source and family with their chemical constituent and uses. Next source we have fruits. Under fruits we have amla, bale, mango, and under seeds we have nux vomica, castor, and mustard, and under bark we have cinchona and quassia. Many times what happens is, like, we might know the source, like for example we might know vinca, clove in our vernacular languages, but we might not know the English name there. And for example, let me give you this example, like vinca flower. Outside of our home we have seen, like, we have this vinca flower, we might have seen but will not be knowing the name. So for your better understanding, here are the different leaves, flowers, fruits, seeds, and barks, and you can see here the images. So there is a trick, like whenever you study any of the crude drug in pharmacognosy, it's better to always study with the images for your better understanding. Drugs obtained from the animal sources. Here are the different animal sources such as insulin, thyroxine, cod liver oil, honey, conchian blood, shark liver oil, and spermaceae, and different sources and their uses here. Drugs obtained from the marine sources. Here the organism is given, their compound, and the chemistry, like which type of peptide bond is present, or whether it's alkaloid or steroid. Next come organized drugs and unorganized drugs. All crude drugs are mainly of two types based on the sources. If the drugs are procured from cellular parts of the plants in raw form, it's known as organized crude drugs, whereas the drugs that are procured from non-cellular parts of plants are known as unorganized crude drugs. Following is a table where there is a difference between the organized drugs and unorganized drugs. In organized drugs, they are the sources from plants and animals, whereas unorganized drugs, they are source of plants, animals, and minerals. Here they are directly procured from the above sources, whereas in unorganized drugs, here they are obtained by extraction, distillation, incineration method. Then they have a proper cellular structure such as leaves, flowers, fruits, barks, roots, woods, etc. But here they have gum, mucilages, and resins. Here they are identified by the morphological characters, and here by the organoleptic characters. They are solid in nature, whereas unorganized drugs may be solid, semi-solid, or liquid in nature. Then in organized drugs, here you can see it can be seen under the microscope, whereas here they have done with the optical rotation, viscosity, refractive index, and chemical test. Examples of organized drugs are woods such as quassia, sandalwood, leaves, digitalis, eucalyptus, mint, and senna. Under unorganized drugs, we have examples such as dried latex such as opium, papain, dried juices such as aloe and quinoa. Stay tuned for the upcoming videos where we'll be discussing the other topics. And once we complete the entire unit, we'll be discussing the important GPAT, NIPER questions and other competitive exam questions. Thank you.